Webinar is now broadcasting. Excellent. So I'm hoping people are joining us. We're at just about 4 o'clock now. So hi there, I'm Fran Sconce and I work at the Royal Entomological Society. So I'm just doing a bit of brief housekeeping for our Verrall lecture. And I'll just move forward, hopefully, to the next slide. Excellent. So this is our title slide. Erica's got a beautiful title slide coming up later on. So we'll start very soon. And the hashtag that we have been using so far is hashtag Verrall. So you're welcome to talk about this on social media. I just want to highlight that Erica has 2 amazing books all about flies. And I can see that you can buy them from the Natural History Museum shop. And I understand that other bookshops are available, but the Natural History Museum has published these books. And I'm sure Erica will talk much more about them in due course. And also published today is our new Royal Entomological Society handbook. This is by Olga Civil, and it's all about blowflies. It's very, very topical for today. So it's available from our website. If you go to our website and go to the publications section and then handbooks, and it's £35, but if you're a society member or fellow and you're logged in, it is £24.50. And then you have to pay more postage as well. And then just to highlight some of our upcoming events, most of our events are online for obvious reasons. So we actually have a young person's Verrall lecture on Saturday with the Amateur Entomologist Society. So if you know any young people who want to hear all about flies in a slightly more accessible way than Erica might talk about today, that's aimed about 10 to 14-year-olds, that is, and lots of other events on our website. And also to highlight, we have a membership. We're a global community. And for students, which is a new innovation for this year, students get their first year of membership for free, which I was so excited about because I think we can reach a whole new audience of people. So some of the benefits of membership you can see detailed here, including eligibility to apply for grants. You get your Antenna magazine quarterly, and you get reduced subscription rates for our journals, our 7 scientific journals. Okay, see if I have any more slides. I don't, brilliant. So I'm now going to stop sharing my screen, and I believe I will hand over to our president, Professor Helen Roy.
Thank you very much, Fran, and thank you for all that you've done to organise this event, and to the Royal Entomological Society and the Entomological Club for supporting this event. It's absolutely wonderful, and I'm delighted to be here today and to be introducing Erica. I'm Helen Roy from the UK Centre for Ecology and Hydrology, and the current president of the Royal Entomological Society, and very proud to be in that role for the Society, and would really like to encourage all of you to think about joining either as a member or fellow or student member. It would be wonderful to welcome you within the Society. But regardless of whether you're a member or fellow, you're extremely welcome anyway. So I have the great pleasure to introduce Erica, and I'm going to keep it relatively short because I'm sure that we're all particularly keen to hear Erica, or maybe you've already heard Erica this afternoon because maybe you were tuning in on Radio 4 to a series that Erica has been putting together for Radio 4 on metamorphosis. So I'm sure we might hear more about that later on, but I'm sure if you didn't catch it earlier on, you can listen to a podcast at some stage later on. So Erica is the Senior Curator of Diptera and also Siphonophora at the Natural History Museum, and her research spans the world. She's done research in Australia all the way through to Costa Rica, and I was just looking through your Twitter profile, Erica, not in a sort of stalking way, but in an interested way, to find out more about what you've been doing very recently. And I saw that you're currently reviewing the Sylphidae of Chile, and then looking forward to moving on to some other books as well. But also you've been very busy teaching entomology as a visiting lecturer for many different universities. So it's really clear that communication is something that Erica has a passion for and just does exceptionally well. And so we're all really privileged to be able to have her here with us today as giving the Verrall lecture. But Erica's also the president of the Amateur Entomological Society, and I was enjoying before we broadcast to everyone hearing about Erica's role there and her enjoyment of that role and the society. But she also has a number of roles within the Royal Entomological Society and is on the Handbook Steering Committee and also a member of the Westwood Medal Panel. So thank you very much to Erica for all that she does for the Royal Entomological Society as well. So it definitely wouldn't have escaped your notice that Erica has been on the radio, on the television, writing books, writing articles, communicating far and wide. And her two books, Secret Life of Flies and The Inside Out of Flies, are both available, as Fran said. And I have before me The Secret Life of Flies, and I was just browsing through it again before coming on today, and I have to see this picture here, and I thought it was particularly timely to show that one and to read the caption that Erica says, "This is possibly the cutest animal on the planet, the grey bee fly." And while I might wish to disagree, because the 24-spot ladybird larva is the cutest animal on the planet, without a doubt, I did think it was particularly timely to mention a bee fly, because this is the time that we're starting to see them out and about, and I can then shamelessly promote the recording of bee flies through Bee Fly Watch, and to share your excitement of bee flies through that particular initiative. So it's very clear that Erica has done a huge amount for entomology and for promotion of entomology all around the world. And I wanted to, though, also say that I think what is really fantastic as well is that Erica's expertise is also matched by her extreme enthusiasm and passion for the subject. And I've had the great pleasure of being welcomed behind the scenes of the museum with the Dipterus Forum. And it was just wonderful to have that welcome from Erica and to feel so at home in the collections, and as a coleopterist to feel very at home with dipterists. So I'm very much looking forward to hearing from you, Erica, but thank you on behalf of all of us for your warmth and generosity regarding entomology and more. And over to you, Erica.
Oh, thank you for that. Coming from a coleopterist, I'm very flattered. Thank you. Okay, let me try and do this. Okay, so hopefully you can all see this.
We can, it's perfect. Thank you.
Okay, brilliant. Right, so yes, I do have a rather good fondness, as it were, an inordinate fondness, some might say, for the flies. I think they're absolutely fascinating, and it was a great honour to be invited to give this talk today, because this is the Verrall lecture. And who is George Verrall? Well, George Verrall is a dipterist. And so it's really nice to bring it back to talk about Diptera. And we've talked about Diptera in Verrall lectures a few times. So what I'm going to do today is just going to give you a romp through the insides and some of the outsides and some of the fun stuff associated with flies in celebration of this great man. I mean, sadly, we would be going for a supper afterwards, but we can't actually enjoy a glass of wine together, which many a fly does. But hopefully you can have some later. So George Verrall was a fascinating man. He was very important when it comes to UK Diptera. He and his nephew described over 550 species of fly. But he also was very interested in writing lists, and he was the first person to actually catalogue how many flies were in the UK. And back in 1888, just a year after the suppers began, he actually put the first list together. And on this first list of species of flies, there was 2,881. This is quite small in comparison to now. We've just moved on another 140 years. And today, or the updated figure I can find, is 7,216. Very thanks to Peter Chandler for updating that. So there's a huge number of flies. And we have been fascinated with them, or some of us have been fascinated with them a long time, looking at them, to naming them, to describing them, to understanding this structure. One of the earliest people to really start looking at their morphology was brought about was this guy called Robert Hooke. He had produced this book called Micrographia, which is just exquisite. Now, he developed one of these. There was the invention of the microscope, and he decided he was going to build one himself. And what better creatures to look at than those around him? Now, people say those flies are a nuisance, they're everywhere, but because they are everywhere and they are in your face, he was able to study these fascinating things. And maybe one of the earliest examples of an incorrect taxonomy, he describes this as a dronefly's head here on the left, which it's not, it's a horsefly, but he's really looking at the structures. And he's the ones on the right, we've got the mosquito larvae. And what is beautiful when he describes these pellucid bodies is that he actually draws the organelles within them. So he's really looking at the features, the structure, and trying to understand it. And obviously, in honor of today, we've got a Caliphorid that he drew. So these beautiful creatures, he was really beginning to think about their morphology and structure, how we could identify them. This happily progressed onto Jan Swammerdam, a great Dutch naturalist, biologist, medic, you name it. He was basically involved with it. And he was also a fabulous illustrator. And here he is. This is the attention to detail I absolutely love, because not only has he drawn the mosquito, he's gone back and realized actually he hasn't done enough detail on the wing. He's properly, again, with his own version of a microscope, properly looked at the scales of the wing and done that. Now he is instrumental, he is pivotal in our understanding of insects and life cycles, because he was the first one to properly explain metamorphoses, which if you listen to Friday's episode, I believe it is, you can understand more about this fantastic man. So he talked about this metamorphosis, and he used these cameras, and so for hundreds of years we've been looking at it. And this brings us right up to date with Olga's amazing new publication. I've been telling her not to tell everyone, it was meant to be a surprise, but obviously everyone blew it. But please go and look at it. And here she's able to use amazing digital imagery, she's able to look down cameras, do all sorts of things to properly look at the morphology of these amazing creatures. It's a great handbook. So why flies? Why are they so amazing? Well, obviously they're amazing, but there's something about their form and their functionality and their ecological range and their diversity that I find truly amazing. And when we think about flies, we often think about the adult stage associated with them. But in doing that, you miss so much of the importance of what these creatures really are. Because the larval stage is equally splendid in its ecological function, in some ways more, but because in so many of the flies that the larval stage is it's just a maggot, or it's a very simple like structure, and how do we understand what's going on? And this is a real shame, because so much of the ecology of the life cycle of the fly is taken up by the maggot stage, because it's predominantly where the feeding stage is. Now, we're seeing this, apart from today, because it's absolutely pelting it down with rain in my garden. But right now, everyone is going out there and we're beginning to see the signs of spring. We've got hoverflies everywhere. Just look on recording schemes right now. And the hoverfly lagoon watch, which is a fantastic thing going on, is going out there and spotting these lovely larvae, because this is a rat-tailed maggot larvae, and it is brilliant in its form, and it loves nothing more than hanging around in dank, dark, sewage-ridden, disgusting environments. There's even some examples of it with myiasis, but I will not go into it because we're slightly too early in the day for that sort of thing. But these are great. And look at the morphology of it. This is able to get into these really dank and noxious environments because it has this telescopic anal spiracle. It breathes out of its bums. It focuses its feeding on the mouth part, the head end, and then concentrates the breathing on the other end. It is an amazing morphological adaptation. And it's great because they get rid of so much of our decomposing material, these maggots out there in the environment. And then when they metamorphosize, when they pupate, you've got these really important pollinators. So the whole life cycle of the fly is really important. But you get some great things. I mean, let's not all say that all maggots look alike. These wonderful faces, which, I mean, come on, they're adorable. Look at their little smiles. Again, we are just looking at the bottom. We're looking at the derriere, the back side of the maggots here. And those eyes, there again, it's breathing spiracles. And here you can see truly that we can look at differences between species. These are in the crane flies, quite simple, quite a primitive, fly. But we can actually use maggots to help us with things like forensic entomology. So because of everyone, I thought best not put anything gruesome. I got sent many a fantastic video by Martin Hall, who is the king of the maggots, but we've just done these. And these are so useful because they can help us with forensic entomology, they can help us with maggot dividement therapy, and they're really good nutrient recyclers. Because these are so plastic in their form, they can get into spaces not a lot of other creatures can get. Which brings me on to my favourite botflies. I, like all good entomologists right now, I have some botflies on my desk in my office, but some of them live in really extreme environments. And maybe, arguably, one of the most endangered species on the planet is the one that lives in a rhinoceros. So here is the rhino botfly adult, and its larvae lives in the stomach lining of a rhinoceros. So when we think about endangered species, everyone goes to the big charismatic ones, but no, actually these poor little amazingly beautiful flies are the ones that need saving as well. So I will get on and talk about the wonderfulness of flies. And so we always start with the head. The head in the flies, as with most of the insects, is where most of the sensory equipment is. Okay, and everyone talks about the flies being absolutely filthy, but they're not. In fact, they have to be meticulously clean, because if any dirt, dust, material, or bit of crud gets in their eyes, it really limits their vision, and they're absolutely essential. They are flying at such rapid speeds, they really need to image, process, and react to the responses around them. I love this picture so much. These are some of the favorite flies. These are pippunculids, and their common name is the big-eyed flies. And I think you might see why we've named them as such. And the reason I've given this picture is not just because it's in a silly pose, and being a dipterist, we're only 5 seconds away from a silly pose, is to show you that some of them are absolutely ommatidia ridden with their eye facets. So the individual eye facets, the ommatidia, make up, and this male and this female here of these big-eyed flies, basically their entire head. Now, dragonflies, we know, have the most eye facets. They can have like 36,000 of them. And at the moment, we only think the diptera have about 7,000. Obviously, we haven't counted a lot of them. But flies are unique amongst the insects in that they do something very different with their sensory pigments within those eyes. So the individual photocell that goes through the ommatidium, that goes through each of these individual eye facets in a fly, is separated back to its constitutional cells. So instead of having one long one, you've now got 8, which is kind of 7. One's fused, 7 and 8 are fused. And so you're able, within the actual eye facet, you're able to get different pictures at different stages. So what does this mean for the fly? Well, it means that they, instead of having just 6,000 images, they can have 6 times 7,000 images. So they can have much greater acuity, much greater sharpness, without having to expand their heads to be huge like the dragonflies. And this makes them some fantastic aerial predators, but it also makes them great mothers, because usually it's the males that do the flirting. But in this instance, it's the female, because she's the one on the bottom here, and she's got these expanded eye facets at the bottom because she has to look for very active hosts to lay her larvae in. So you've got these adaptations going on. But obviously, if we're going to talk about advanced predators, let's talk about some of the biggest or littlest. So here is a beautiful little robber fly, adorable predator, highly venomous, with amazing eyes. And because of this advanced way that they can see, even something as small as that, and this is a finger of Will Hawkes, who posted this image on Twitter, even something as small as that is a really good predator. Flies don't use their eyes just for seeing. No, there's a lot of flirting going on. And again, that's one of the lovely things about flies. They're only just a gnat's breath away from a flirt. The mouth parts is something we talk about a lot, because a lot of you would have come into very close contact with mouth parts of flies, but not all of them have them. This adorably fluffy hirsute creature is a botfly, and it is a reindeer botfly, and it is brilliant. But basically, they do not have mouth parts. They have tiny vestigial mouth parts, and a lot of them at most will just take sips of water. Now, what's really interesting is some research that's come out of the museum using CT scanning and all sorts of advanced ways of processing imaging and looking through the puparia is able to see what's going on. And this is what Daniel's been doing here. He's been looking at what's been going on in the puparium of flies, and he's been able to see the huge guts, because it's just obviously the larval stage that's feeding. It's gorging itself in the in the nostrils of that reindeer or camel, or in the skin of humans. It's gorging itself completely. But during the pupal stage, its stomach, its guts completely disappear to this absolutely vestigial part in the newly emerged adults. They no longer need to feed. But we do have very long mouth parts. I am obsessed with mouth parts, apparently, and an example here is beautiful nemestrinid. Here is a beautiful fly found in South Africa with a mouth part that is the longest mouth part in relation to its body of any insect. I know I went on about the hawk moth mouth parts yesterday, but this one I think is extraordinary. And it is, if it was a human, it's 6 meters long in comparative. Our tongue was 6 meters long. So it's great. But this is used for pollinating plants. This is co-evolved. It's a guild pollinator. It's co-evolved specifically with a lot of flora in South Africa. But that's not the mouth part most of us will know in the UK. Here we go, a little plug for the Bee Fly Watch. This is one of the beautiful Bombilius major, and they are out now. So please do get recording. I'll put a link up there. But it is this lady that is the one that causes the most concern about people when it comes to Diptera against the flies, the mosquitoes. And it is only because she is a poor mother desperate to ensure that the offspring have the greatest chance, the greatest ability to survive, that she needs this blood meal, this protein-rich meal. And her mouth parts are a series of stylets, a series of straws, say, and that she will penetrate most of the skin with them. But what is fascinating about them, and we're only beginning to just really properly look at this, is that these can be bent, they can be manipulated within the surface, and at the very end of her skin to manipulate her mouth part to get to the best part possible. We move on to the thorax. This is a really most likely to be the chunkiest part of the body. This is where we have a lot of the structures associated with locomotion, but we also have lots of bristles. In fact, bristles all over it. This is a new species that my colleague Nigel White and I are describing, and this species is tachinid from Dominica. And these bristles, within the bases of these bristles, there's going to be sensory pits, and these can have mechanical receptors, chemoreceptors, thermoreceptors that are all really useful when it comes to locomotion. And the legs are what they do primarily for the on the ground locomotion. Well, I say the legs are used for locomotion, not always. Lots of legs are used for flirting. Once more, here on the left is a beautiful Dolichopodid from the UK. In fact, this is a very rare Dolichopodid found along the Thames estuary, and it's definitely something you should be looking out for. But it has legs that have been described as having fantastic tangle of ribbons and bristles and hairs and protrusions that defy belief. It is true. What on earth are all these structures for? And actually, that's what we say with a lot of things. What on earth are all these structures for? We don't know. The one on the right is a new species of housefly. Again, we describe them from Dominica. This male has incredibly hairy legs. I mean, it basically looks like me after a month of doing field work. But what are all these hairs for? We don't know. But we do know that some of the hairs are for flirting, and it's not always down to the males. Here's a female, a beautiful empid, and she's got really hairy legs, and she will often wrap them around our abdomen to show off that she's really good. She's going to be nice and fecund. But this is another empid on the right, and this is a male, and he's extraordinary in the fact that his front legs have got expanded tarsa, and within there are silk glands, and he uses these to make nuptial gifts for the ladies. What a nice gentleman he is. We move on, and probably the most recognizable feature of the fly are the wings. I mean, they're called flies after all. And as they emerge from their larval stage, their pupal cases and their wings establish, they become some of the most important aerial flies on the planet. The hoverflies are amazing aeronautical engineers. The fact that they can hover, which is really quite impressive, is truly something. And we're also now beginning to understand not just they're important in pollination, but also how far they travel to pollinate. And this again is really important about the flies. So we get a lot of migrant hoverflies. Will, who I was talking about earlier, is actually working on a lot of migrant hoverflies coming over from North Africa. And the recent paper they put out looked at the aerial fauna and estimated that over 4 billion hoverflies were flying over every year. That's extraordinary. And think about the importance of this. Think about how high they are up in the aerial plankton. They're able to see relic populations of plants and so transfer pollen, therefore transfer genes, therefore help the environment. And they themselves will actually come over here and find our local populations of these and transfer their own genes. So they're bucking the trend when it comes to insect species loss, and they are also helping to provide for a better environment for a lot of the wildflowers. Not all flies have wings. I know, I love this. There's a lot of flies that don't. This, honestly, this video just fills me with glee every time I see it. This is an adorable bee louse. I prefer to call them bee riders. And actually, this image on the left is I've got sitting on my desk here right now. So these are wingless flies. They're basically blind. They've got no halteres. They've kind of taken that body print of what you think a fly is and laughed in the face of it. And what is truly amazing is that we used to think, you know, that they'd been wiped out from mainland UK with the introduction of miticides because of the Varroa mite. But I'm quite happy to say that we've definitely got some alive in colonies around the UK. In fact, 4 sites now have turned up with these bee riders. So hopefully a new publication is going to come out with them shortly. But these were once thought as bad. They were once thought as really bad parasites on the bee, but we kind of know that they're more kleptoparasites. Okay, they would go down to the mouth parts, and when they are swapping food, that's when they steal it. I kind of think this would be really good if you had a beard, and you know that you could train them in your beard to go and clean up all the muck. But a paper that came out a couple of years ago has been like, well, how is this enabling it? I mean, this is quite a large animal roaming around the surface of another animal. That's like a small child roaming all over me. What is going on? And they realized that these little things are mimicking the chemicals released by the exoskeleton of the queen or whatever bee they're on. So this is really interesting because we're starting to understand how they can do this, and this may again have implications when it comes to pest management of other species. I have an adorable, absolute massive fondness for these flies. Okay, these again, we've got wingless flies. Again, these are parasites, but these are parasites found on bats. And I not only like them because of their morphology, I mean, look at them. The ones on the left, we collected off bats in Dominica, 11 o'clock at night, holding angry bats, trying to chase what looks like drunken spiders running around these bats. The one on the right is actually in the Wildlife Photography of the Year exhibit, and it's lovely to see this photo got recommended. Poitra image is truly amazing. And these are two, well, we're calling them 2 families of bat flies at the moment. They may actually be one, the Streplobilids and the Nycterabies. And what is fascinating, not only about their morphology, but they give birth to live young. We don't see this in many insects. It's quite an unusual trait, and it is amazing. So these blind, wingless, they've lost all their muscles in their thorax, but she will nurture her larvae. And they don't give birth to many larvae because it takes an awful lot out of them. But she will reverse off the bat onto the wall or the tree, wherever the roost is, and she will lay this. Basically, it's about to pupate as she's given birth to it. So she feeds it internally. She produces a lactate that it is able to feed upon. So they're truly fantastic. And what is good about them is that the founder of this entomological society that organizes the Verrall Supper, George Samuel, is he described that family. So going right back to the beginning, he was the one, and he actually wrote this fantastic book called in 1819 called the Entomologist's Useful Compendium, or the Introduction of Knowledge of British Insects. And he noted during this that actually insects have been the most overlooked in researches for knowledge. Now, I would argue actually he could just ignore the word insects and put Diptera, because there is still so much we don't know about them. We have favoured many of the Lepidoptera, the Coleoptera, and these other species, but still within Diptera, there is so much more to learn and so much more to see. We end with the end, as it were. We will talk about the final stage of the fly, the bottom, the abdomen. And this isn't often as ornate as, say, some of the legs or some of the wings or some of the headpieces. This is a rather cute Stratiomyid, a soldier fly, with its larger than life bottom. It is the Kim Kardashian of the fly world. But what's in the abdomen is really important, because this is the raison d'etre of why all species are here, is to replicate. And as anyone knows about Dipteris, we are always talking about genitalia. Now, more often than not, we talk about the female genitalia, because this is the male genitalia, sorry, because this is often the most important diagnostic feature. But we should be very careful not to ignore the females. Ignore females at your peril. There's a life lesson. Because a lot of their ovipositors, their egg-laying tubes, are really important characteristics. This is, for example, this is a wonderful Tefritid, and this has this beautiful egg-laying tube. And what she does, she will probe through to lay her eggs in. And again, at the end of these have got sensory cells that are able to actually figure out what is going on. Is this the right environment for her? A lot of the others, there's an ant decapitating fly that she will use her ovipositor to slice through an ant to lay her egg in. And again, our own canopids, our bee grabbers or wasp grabbers, the females there too, she will use her egg-laying slicing apparatus now to slice through the backs of bees and wasps to lay her eggs in. But I guess we have to talk about the males. And here we go, because they do genuinely have some extraordinary structures. I love this. This is a Nature paper on Diptera genitalia. I mean, nothing can get better than that. And we have here some of my funniest examples. I always think about this. These always remind me of the doctor's surgery, when you have that buzzing machine and you have to get round without hitting it. And they so the Aedeagus, the penis of the fly can be massive in comparison. We've got here up in the top right, this is one of the Megacillia. This is a phorid. It's an undescribed species, but look at it. It has a corkscrew. Now, as a female, I don't think that's particularly pleasant, but this does allude to the arms race that's going on between the sexes, because there's a whole lot we don't understand. We're only beginning to look at with this cryptic sexual selection going on. Now, Drosophila are one of the model organisms of understanding genetics. In fact, it was talked about today on the radio, I believe, by a very familiar person. And one of the things we are doing is we're using them to look at the competition between these species and understand what's going on. Now, the reproductive structure is where we finish it, but this isn't always where it ends with flies. In fact, they do some really quite odd and really quite crazy stuff afterwards. This, I'll just leave you a second to stare at arguably one of the weirdest, most extraordinary flies you will never see. This paper came out many moons ago, and I haven't seen anything like this. Yes, the one on the left is an adult fly. Okay, she is an Ascodistron. She is an Africanum. She comes from Africa, and this paper came out in the 1930s. And it actually describes what we're seeing further back, 1912, over 100 years ago. And these bat flies, these are the strebs that I showed you earlier. So yeah, they're a bit weird, but some of these streblids, some of these bat flies are actually born with wings. When they pupate, the males and females look pretty normal for flies. The females have wings. But after she's copulated, she's no longer happy with these flies. And she has this enormous proboscis. And what she does is she sticks her head into the side of the bat. And during this, she rips her wings and legs off. Brilliant. And she snuggles in. And so that basically all you're going to see is the posterior end, that happy, smiley little bristly face that we're staring at on the right. Now, not only does she do this, her abdomen at this point, she invaginates, she folds over the rest of her body. So you just get this extraordinary flask-like structure left. It basically becomes encysted in the side of a bat. So all you do have is this posterior bit poking out. So the morphology of these creatures is amazing. But why is it important that we look at it? Well, I know I'm talking to a room of taxonomists, or thousands of rooms of taxonomists, and I know you understand that, but actually so many people are still getting it wrong, and this is not helpful when it comes to understanding the ecology, the behavior, our environment, diversity, etc. So here we go. This is one of the classic examples of a morphological error. And again, why flies are always getting blamed for doing stuff they're not actually responsible for. Here we are. The fly is even called the devil, which I think is a little bit extreme, but Hymenopterus. Actually, it's your fault, I think you can see here. Thank you very much. And yes, one of my favorite things is to scour the internet looking for taxonomic fails. Now, some of these can be quite amusing. You get these Frankenbees, they talk about the danger of that. I didn't realize hoverflies were so problematic. You get complete identification errors. Diptera larvae suddenly become beetle larvae, which is quite cool. But also, it can be quite problematic. The mosquito here is one of the examples I will use as something where if we get taxonomy wrong, actually this can become dangerous, because instead of actually looking for species that may be damaging to us, we're looking for species that are completely harmless. In fact, these, adult mosquitoes, adult craneflies at best, would take a little bit of pollen, take water. They have no interest in our blood at all. So this can be quite bad. So many times flies are confused with bees. So many times we see this, and which has probably led to the most comical of errors, which will always be highlighted by everyone, especially Dipteris. And the thing is, this isn't even a hoverfly. This is one of the house flies. This is how annoying it must be for the authors of that book. So we really diversity, there's so much we need to know, but there's also so many numbers.
In her 2017 work, The Secret Life of Flies, the entomologist Erica McAlister estimated that for each person on Earth, there are how many flies? The figure in question is about the population of Zambia or the Netherlands. You can have 3 million either way.
6 million?
No, it's 17 million. Amazing, isn't it?
Yes, absolutely amazing. Thanks for that. So what I'm asking you to do is to go out, explore. We've got bee flies about to hit the garden. We've got hoverflies everywhere. We've got recording schemes set up in the UK. We have the Dipteris Forum, which is an amazing organization. We have research going on within the museum. We're trying to understand the whole diversity thanks to the Darwin Tree of Life project. We have all these different ways that we can get involved, start looking at flies and understanding more about them. We have new species we know in the UK to describe, and we also have some very important endemics only found in the UK that need looking after. So I would ideally like to raise a cheers to all of you in person, but instead I would just sit in my shoffice and maybe raise a glass of a fabulous gin and thank all the flies that have been used in making this slide and all the people that are helping me. Thank you, the Royal Society, for letting me do this.
Thank you so much, Erica. That was just a fascinating and whirlwind tour through the world of flies. Just absolutely wonderful to see such diversity and to be taken through it, through the morphology of these just incredible insects. Just a really, really wonderful talk. So thank you so much for that, Erica. I'm going to go straight to the questions because there's quite a lot coming through for you. So I hope that's all right if you have time to pause and take a breath. And
I won't take the gin yet.
Don't take the gin yet, no, or the beer, or the combination of the two.
Okay.
I'm going to now coordinate my stuff across the. We've got some questions coming in on the question. Please, if you've got further questions, please put them on the Q&A, but there are also a few on the chat at the moment as well. So I'll just scan across to both these. There's lots of people saying, thank you very much for a great talk. And I guess in some way makes up for our lack of being able to give a round of applause or a standing ovation at the end of your talk. But hopefully you can see through the chat quite how much appreciation there is for your Verrall lecture. So congratulations on that. I'm going to scan up to one of the questions that's in the chat because I think it's a wonderful way to start the questions, because someone was asking, given that there are flies that don't have wings, how do you then go on to define what is a fly?
Welcome to our world. So there will be, I mean, there are other features that you will know which are associated with flies, the adult stage of the fly, should I say. So we will look at the mouth parts, we will look at sibling sisters, we will look. Often we know the shape. I remember the first time I came across wingless phorids in Costa Rica, and I'm staring at this pot and I'm like, I know you're a fly. I absolutely know it because I know the form of you. I know how, you know, the arrangement, the antennae, which is again a really important feature. I know you're a fly, and actually I know you're a phorid, but what are you doing to me? And at that point we can go away and start to identify it from either specimens in the collection or associated literature and things like that. Oh, that phorid.
I love a phorid, I have to say.
To be fair, I do. They're adorable. Ecologically, the most amazing family of animals on the planet. They eat absolutely everything. I love the fact that some have specialized in eating shoe polish. I mean, come on, flies. What are you doing?
Some of them parasitize ladybirds, which is, of course, the most fascinating of them all. So we have a couple of questions that come in very well and nicely together. One is, what is your favorite fly or your favorite group of flies? You can have either one.
Oh, this is, I'm not a mother, but I guess this is what mums think, isn't it, when they get asked about which is your favorite child, because you probably do have one, but you wouldn't admit it. So I do love the more bitey stabby ones. They are, you know, I mean, who doesn't? So robber flies I just adore because they're highly venomous, amazing predators. They are so active, and we get some really good species in the UK. 2 years ago, it was the first time I was able to see the hornet robber fly, and I danced and screamed. It was so exciting seeing that. But when it comes to cuteness, I guess it's either the bots or the bee flies, because they're so hirsute. They are, you know, when people go on about a charismatic mini fauna, they're the first things that spring to mind with them.
Yeah, no, absolutely, they are beautiful. This is probably an even more difficult question then, and then we'll get onto some of the other more detailed questions. Someone is asking, what is your least favorite fly group, if you like? There isn't one. There is no answer to that question. We have come up with a question for which there is no answer.
I don't know.
Well,
there you go. That's my really. The thing is, I'm really lucky. We work at the Natural History Museum, and we have visitors from everywhere. And you can't help but just be inspired every time they come in and they're looking at a new species, or they're looking at the collection, and they tell you the most fascinating stories associated with them. So you think you don't like one as much, and then you're like, oh, but that's very interesting. I like that. And so you just get sidetracked.
yeah, no, I feel very much the same. There's always something positive to say about an insect, regardless of what that insect is. I think they're all absolutely incredible. I just want to say as well that there's quite a number of families watching your talk and all saying how very much they're enjoying it. And so just to highlight again, there is going to be, Erica's going to be giving the Verrall for a younger audience. So you'll all be very welcome to watch it again when Erica delivers that. So I'm going to now go through the Q&A. Please do keep posting your questions, and we'll just get through as many as we can. I'll just go through in sort of sequence. So Megan asks, are the rhino botflies specific to the species or subspecies of rhinos?
Yes. So there was 3 species of rhino botfly. So there was the Javan rhino botfly that I believe was last collected in the 1850s, something like that. There was another species of botfly, which rhino botfly, I've forgotten. And then there is this one that I showed you, and that is it's in the northern and southern. It is in black and white rhinos, but it was not often collected. It comes mostly, we get from zoo animals and things like that. So it's it's just a sad thing. But with the Javan rhino, I mean, or Sumatran rhino, that's what we're really worried about. It's a poor collecting habitat. So we may not be reflecting true numbers because of, I mean, ideally I'd like students to go chasing around after rhinos immediately they've defecated, but health and safety decree that's not probably ideal.
No, it is fascinating though, and I think it does really highlight how, as numbers begin to plummet for a particular species, the interactions that that species have with others also then plummets as well. So that idea around sort of extinction of interactions, I think that really sort of brings that out as well, and the importance there. So someone is also asking, obviously botflies are causing lots of excitement this afternoon. Is the cycle the same for all botflies, i.e. the decrease in digestive system?
No. So you have 3 different types of botfly, as I affectionately call them. The human botfly is subdermal. So this would be underneath your skin. And what's amazing about the human botfly is because it's quite a noisy fly, it will actually catch midair other species which they will use as vectors to transmit their eggs. So we have got species of mosquito in the collection that have got botfly eggs on them. So the adult the mosquito. So you could get a mosquito bite, as it were, and a botfly, which is quite entertaining. You have the rhino ones, which are the the stomach lining ones. And then the favorite ones, the reindeer ones and whatever, I affectionately call snot bots because they live in the nostril. And I've got some camel. This is the camel botfly on my desk, which is the silliest name ever, Cephalopus titillator. Which, so apparently it was titillating the nostrils of a camel, which I very much doubt somehow.
Thank you very much, Erica. So there is a question from Jason Chapman asking, what group of flies do you think a budding young naturalist should choose to focus on?
Right, so here we go. There's 2 groups or 2 sets of groups, and I can't find the other book, but the first one, I would just go hoverflies. Really nice, simple keys. A lot of them can be done pictorially, so it's really easy for you to get in there. The UK hoverfly recording scheme is up, it's active, it's amazing. There are so many people out there to help. And the second group of flies is the soldier flies and their allies. Okay, again, I don't know what I've put my book. I think my computer's leaning on my book. So this, again, really good key, really useful, really nice online links. In fact, there've been some really good pictorial links that they've done in the last year to help you identify all the differences. So again, these can be done a lot with images, and it's really good that we can add really important data recording schemes.
That's fantastic. And just to say as well, on social media as well, there's a lot of dipterists who are really active and very happy to help with identification, as well as Erica and all of her team in the Natural History Museum as well. So that's fantastic. And just going through a slightly longer question here is about how flies perceive their environments in relation to light, and if they are blind and have no eyes. And a report that C. Vacinia with surgically removed optical lobes was still able to demonstrate photoperiodic reception. How are they able to do this? Do you know how the blind and eyeless flies can detect and utilize light cycles, etc.?
Not specifically with that. I should have read that. But what? So I know with larvae they have photo cells under all their skin. So if the back end of a maggot is poking out in the light, it can detect that, even if the head end isn't, which is it's really amazing. We get a lot of flies that are living absolute darkness. In fact, the largest, there's a blind flying fly that is a true troglodyte. It never leaves its cave, and it's very hirsute. So it picks up a lot by its sensory by that way, mechanical sensors. A lot of them do fly at night. In fact, so many of these cryptic species is because they're flying at night when entomologists are imbibing or in bed. And so we're only just beginning to understand how they're able to see. And, you know, they have really good light. I'm a fly. That's me showing you the ocelli. So the ocelli at the top as well, they're able to pick up the light and dark pigments as well and help stabilize it. So they have got all these different receptors, and probably a lot more that we don't know about.
Yeah, no, I think absolutely that the sort of sensory side of the ways in which insects are living their lives is kind of beyond our imagination. And there's certainly so much to, when we think about what the Lepidopterists are finding out about the sort of night vision, it's just incredible.
Yeah.
There's a question about what's the benefit of 2 wings as opposed to 4? Why only
Well,
have 2 wings?
why not? You have got, I mean, why not? They over time, the second pair of wings has become these halters, these amazing balancing gyroscopic proprioceptors, and these are really fast acting, okay, and they're very, very good at moving. They've got resilient, the elastin protein in their base, and so they will shut back into shape quite quickly. And a lot of the time as well, they will be permanently moving to ensure that the muscles are warmed up and ready. So you have this kind of coiled fly. So when you try and swat it, which you should never do, it is already active and ready to go. So they have a really amazing maneuverability as well. We're cleverer people than me. I'm not that. Are looking at how we can develop technology, jump jet technology greater than it is, based on these flight mechanisms of flies. And because of the halters, they're able to stabilize themselves. I mean, they can roll, yawn, pitch, they can do all of these amazing things, which you're not going to find in all of the other insects. I'm not going to say all the other insects. It's a lot of entomologists there.
Thank you, Erica. We're going to go straight from the wings to the genitalia, which I'm sure is going to excite you no end, Erica. So regarding the corkscrew external genitalia from the males of the species which have those corkscrew genitalia, what is the female receptive tract like? So there we are. We're getting straight to the nitty gritty here.
Well, the thing is, so we don't really know because this megacelia, even though Brian's the world expert on them, he doesn't know what it is yet, let alone explored the female, as it were. But we're only beginning to look at the female genitalia. I mean, the thing is, it's internal. And so thanks to things like the computer tomography, now we've been able to explore and have a look. Now we do know in things like Drosophila, where we have been exploring, that their ovaries, the ducts as well, are extremely long when they've got such weird structures. Drosophila, not only is its genitalia weird, its sperm is weird. So it's got the megasperm. So the Drosophila melanogaster, its sperm is 1 millimeter long, but the bifurca is 5.8 centimeters long. And that uncoils as it goes in. And she's got these hugely developed internal structures to enable this competition to going. So as he develops more, she develops more, and so and so. So we are beginning to look at these. I'd love to get some grants to go scanning all through the genitalia of the museum's fly collection.
No,
it's a weird one to write.
It's really fascinating. Evolution is a wonderful thing. There are a few questions around flies as food for humans, and whether you would like to say anything about flies as food for humans, and just a follow-on question around using flies as food source, and given that the larvae eat some fairly disgusting material from our human point of view, the person Judy Staines clarifies, would the larvae or adult, whichever used for food, be safe for consumption?
Yeah, in fact, we've got factories all over the world with larvae of flies actually occurring now. So the one we are using at the moment is the black fly, Hermatia hallucens, and this is a really popular one. I believe there's 10 large factories going on in South Africa, but I could be slightly out of date now. And this is amazing because, yes, it is feeding on decomposing waste, but this could be like kitchen waste. So you can have your own compost being fed upon by these flies, that one day in the future, you could be munching on the larvae or the pupa of that flies. Now, you'll be glad to know that we do not feed on any insects that's fed on waste produced by animals, because our food standards will not allow that. We've had few histories with bad eating of food, and so we're very, very prescriptive about that. But insects are weird. They're not in the phone. We just don't know how to describe them when it comes to eating, which is ridiculous because 2/3 of the planet eat insects anyway. We eat over 2,000 different types of insect already, so it's not unusual for us. Housefly larvae we've been looking at as little protein packs to feed livestock, etc., because they are jam-packed with omega-3. They are so good with protein. In fact, like for like, the black soldier fly and the houseflies, they have more protein in them than say salmon and things like that. And so not only is this going to be good for the animal, it's going to be good for our environment because they don't need water, we're also going to save a lot of other species. So hopefully this is going to move on faster. I've got some fly flour in my cupboard, and so I mean people get a little bit sensitive to eating insects, I don't know why. So the idea of making it into these secondary products like pasta, which is quite nice. I'd like some fly pasta, please. So not a fly in your soup, it will be actually a fly in your pasta, which is quite nice.
That's fantastic. Just going to check with, we've got more questions coming through. Are we okay just to keep going? Are you okay to keep going, Erica, for a little longer with some questions? There's lots coming through, and somehow or other your fly pasta response takes me back to Sonia's question about the corkscrew genitalia again. So it's all becoming...
It's opening the wine. Multifunctional. You can use them to open the wine and then eat them afterwards. How great is that?
John Baird has a question about Diptera larvae tend to be legless. Have you seen any leggy or pro-leggy Diptera larvae?
There are some. So it is in the, what's the family? It's one of the Brachyserian families. They, gosh, can't remember it off my head. They have got some nearly legs, but we do have a lot of pro legs. So chironomids and things like that. So you in the Nematossaran fly family, so a lot of the aquatic families, there's some really beautiful, like the Blephasteridae and things like that. They just look like tiny little kind of aquatic woodlice in many ways. Like imagine that with suckers on, and they were sucker along the ground of a fast running stream. So you get some really quite funky looking things there.
Fantastic. And there's a question here from Sarah Arnold, which links really well with the fact that at the moment the Royal Entomological Society is undertaking a project around grand challenges in entomology, which is inviting everybody to come up with lots of ideas around the grand challenges in entomology. And Sarah's asking, have there been any recentish cases where you've come across a fly specimen or specimens in the Natural History Museum collection where on closer examination of it, you've learned things that have really changed our understanding of Diptera or wider ecology?
Me per se, not as such, but there's some questions I really want to ask. That's where I would like to do, and is using some of this novel and new technology to answer questions. So I'm working, say, on novel technology, looking at historic DNA in mosquitoes, and that's great. We're looking at old specimens, and the museum is full of old specimens, and it's like, why do you need them? There's many reasons we need them. And one of the questions I would like to ask out of all the flies is to actually look at their gut contents. And I don't want this for just the medical reasons. We don't want to just analyze the blood, but what about the pollen? Because if we can start looking at the pollen associated with their mouth parts and their gut contents, we can properly incriminate what they're pollinating. And this will have a massive impact. So we have, you know, hundreds of thousands of specimens, millions of specimens in the museum that are just little data packages waiting to be explored. And if we could start doing that, if we could say we can look at the genetics of that fly, I could tell you where that fly came from, I could tell you what that pollen is doing, imagine how important this would be.
Yeah,
there's a NERC grant there, like massive science grant. Please give us loads of money. That'd be lovely.
You know, it's really fascinating, and most definitely museums are just such a fantastic resource of information in so many different ways, and each of those little specimens holds so much information that's absolutely incredible, and including some of them with the parasites, I'm sure, alongside the hosts.
We have them with parasites on them. We have them with associated food. We have all these different types of information that has just been held there, and I mean, it is active. It's not you know, people talk about them being static collections. They're dynamic. We're working on them. We're using them. We're building them. Our knowledge is just getting more and more every time we look at them.
And most definitely, it's just always so wonderful to be welcomed behind the scenes, whether in the
one day,
the Natural History Museum in London, or the Natural History Museum in Oxford, or indeed in Edinburgh at the National Museum there. It's just so many welcomes around all of our museums across the UK. So thank you to you and all of the curators who make that possible. I think we've still got more questions coming in, which is just absolutely wonderful, but I think I should ask the last question. And I'm choosing the question from Alex Hyde. Do you have ambition to play host to a botfly larva?
Oh gosh, yes. Oh, come on. This is like every dipterist's dream. Come on. So of my colleagues, I think 3 of them have had them. I've got some lovely stories of people I've worked with who've lost their larvae on a plane, which they were coming back from Costa Rica, and they were going across to Thailand, and they knew they had it on them when they got on the plane, and they couldn't find it when they got off, which would have been a great thing to clear up. But yes, no, and there's a brilliant blog piece about an American dipterist who reared up his own one, and the images. I mean, he had a beautiful, pristine adult at the end. Can you imagine adding that's your legacy in the collection? I've been donated larvae from various people around the UK, but no one has donated me an adult yet. I'm just waiting for someone to do that.
Well, thank you very much, Erica. And there are still more questions coming in. And I'm sure if you'd like to ask your question to Erica and you're on social media, you can send Erica a tweet or indeed get in contact through the museum. But just to thank Erica for an absolutely captivating hour of flies. It's just been really wonderful. And the breadth of subject material that you've covered and the wonderful images that you've shown to us, I'm sure will stay with all of us. And it's just such a fantastic celebration of today being the Verrall lecture and being on flies. It just makes it extra special. And so to thank Erica and the Entomological Club and the Royal Entomological Society for making today possible, but also thank you to everyone who's been posting such fantastic questions and so much support for Erica's work. And it's been wonderful to hear about the bee flies just getting up and about now and get involved with their recording, but also that shout out to museums and the wonderful role that the collections play in terms of natural history and far beyond. So thank you very much, Erica. We've all been absolutely inspired, and we're looking forward to hearing more from you when we switch on the radio or the television, or indeed when we hear more talks. So just thank you to Fran as well for organizing this and making everything go so seamlessly well. We hugely appreciate that. So it just leaves me to hand Erica the President's Medal. And I wish that we were together in person, that we could go and have a drink together afterwards and a good chat about ladybirds and flies and all things insects. Will be thoroughly inclusive. But let's hope before too long we can all be together, and let's hope this time next year we'll all be heading off to the Verrall Supper together. So thank you, Erica, and thank you to all the wonderful entomologists who've joined us today. It's been great being with you all, and I get to present to you the President's Medal. So here it is for you.
Now look at the magic of that.
Wow, we did that
Amazing.
seamlessly. That was amazing.
Thank you.
An absolute pleasure and an absolute honor to be the person to introduce your Verrall lecture. And thank you very much again, Erica.
Thank you. Cheers. Thank you, everyone.
Thank you, everyone, for the wonderful questions as well.